day everyone, this is Dr. Nica and this lecture video is a continuation of the pentose phosphate pathway lecture. In the earlier video, we discussed the oxidative phase, and at this point, we are going to be discussing the non-oxidative phase, which we have mentioned earlier, that is the reversible phase. So I have already given you a quick background of the non-oxidative phase in the earlier lecture, but right now we will look at the different steps one by one, and after which we are going to discuss what are the functions of NADPH in the pentose phosphate pathway and what are the clinical correlations if this is not sufficient enough for the body. So we have here what we call the non-oxidative phase, which is the reversible part of the phase. The first one that we're going to discuss is the epimerization. So we have the ribose 5-phosphate epimerase. So this will convert ribose 5-phosphate to your cellulose 5-phosphate, and the mechanism is known as epimerization. So this reaction will flip as the stereocenter in the substrate, and an epimer of a compound occurs when only one stereocenter is flipped. So in this case, we have upon epimerization of ribose 5-phosphate, it will be converted into your cellulose 5-phosphate. Okay. So here in the next slide, we emphasize on transketolase and transaldolase. So these will compose of a myriad of steps that are required to transform pentoses, so ribose 5-phosphate, back to glucose. Okay, but back to glucose, so glucose 6-phosphate. So besides the epimerase, which is here, only two other new enzymes are needed. So what are those enzymes? We have transketolase and transaldolase. So we'll start first with your transketolase. Your transketolase is actually an enzyme that uses a cofactor. As we have mentioned earlier, a cofactor is a non-protein substance that helps in the biological chemical reaction. So what is the cofactor? So you have to remember this class. Transketolase, its cofactor is thiamine pyrophosphate, and for purposes of discussion, we are going to use TPP to mean thiamine pyrophosphate. So here it is used to stabilize a 2-carbon carbanion intermediate. So we can see here from cellulose 5-phosphate, after epimerization, it will be converted into glyceraldehyde 3-phosphate. As we have mentioned, this is an intermediate of glycolysis. Okay. We have here also what we call transaldolase. So this enzyme is a protonated Schiff-based intermediate with a ketose. So it stabilizes a 3-carbon carbanion intermediate, which now allows an aldehyde-based sugar to react with enzyme-linked ketose. So the mechanism is similar to that of aldolase. So from glyceraldehyde 3-phosphate to erythrose 4-phosphate. So this is resonance stabilization. Now, having seen this and having understood what is the oxidative phase and non-oxidative phase, now let's discuss class. Since pentose phosphate pathway produces NADPH for the body, we have to understand ano yung function nito. So first, we have here a reductive biosynthesis of fatty acids and steroids. So just a quick review. When we talk of NADPH, we talk of NADP plus and how it differs from NAD plus. So it only differs by the presence of a phosphoryl group, okay, as one of the ribose units, which now allows NADP plus to interact with NADP plus specific enzymes that have unique roles in the cells. So when we talk about class reductive biosynthesis, what we mean is that this NADPH actually is a high energy molecule wherein its electrons are destined for use in reductive biosynthesis rather than for transfer of oxygen, as in with the case of NADH. So meaning in metabolic transformation in the hexose monophosphate shunt, part of the energy of glucose 6-phosphate is conserved in the form of NADPH And this molecule can be used in reactions requiring a high electron potential electron donor. Example, NADPH now is used as a source of electrons for the biosynthesis of fatty acids. So, reductive biosynthesis. So, it is required for us to be able to synthesize fatty acids in our body. Okay. In the next slide, we can see here that it is important for glutathione reduction inside the red blood cells. So why do we need to understand, why is there a need for glutathione reduction? So first we have to discuss the reduction of hydrogen peroxide inside the red blood cells. And why do we need this? So hydrogen peroxide class is one of a family of reactive oxygen intermediates. So these are oxygen intermediates that are formed from the partial reduction of molecular oxygen. So byproduct ito. It is a byproduct of aerobic metabolism. So hydrogen peroxide is something that the body produces after nagkakaroon tayo ng aerobic metabolism in the cell. So it is highly reactive and can cause serious chemical damage to DNA, proteins, and unsaturated fatty acids. So meaning we need to reduce hydrogen peroxide because it is detrimental to the body. Now, hydrogen peroxide, which is in reactive oxygen intermediates, they are implicated in many different diseases. So we have reperfusion injury, cancer, inflammation disease, and aging. That's why it's important for us to have these reactive oxygen intermediates to be reduced. Okay, now, meron tayong several protective mechanisms of the cell, and it includes reduced glutathione. So, first, the enzymes that catalyze antioxidant reactions is called reduced glutathione. So, pag-usapan natin kanina yung reactive oxygen intermediates, they have to be reduced. So, they have to be catalyzed, and these antioxidants has to be catalyzed using a reduced glutathione. Itong reduced glutathione is present in most of the cells. It chemically detoxifies hydrogen peroxide, and this reaction is catalyzed by glutathione peroxidase, which forms oxidized glutathione. So this oxidized glutathione no longer have protective properties. So we have to regenerate it kasi na-oxidize yung glutathione peroxidase. So the cell will regenerate reduced glutathione in a reaction that is catalyzed by glutathione reductase, which needs NADPH. So kailangan yung NADPH in order for us to regenerate, or in order for the cell to regenerate reduced glutathione. So in the absence of NADPH, we will not be able to produce reduced glutathione. Therefore, these antioxidants will not be catalyzed. So NADPH is the source of reducing electrons in this process. So therefore, we can say that NADPH indirectly provides electrons for the reduction of hydrogen peroxide. Now, it's too much wordy yung pinag-explain natin earlier, and we'd like to correlate this clinically by showing you this picture. So, reduced glutathione, or GSH. So, here we see that is the reduced glutathione. So, it removes hydrogen peroxide, H2O2, in a reaction catalyzed by glutathione peroxidase. So, reduced glutathione is regenerated by glutathione reductase, which requires NADPH. So, as we can see here, protection from radical damage. So, we have here oxygen. So, upon mitochondrial respiration or radiation, sulfa drugs, herbicides, and anti-malarials or divisin, nagpo-produce siya ngayon ng superoxide radical. So, ito na yung sinasabi natin na reactive oxygen intermediate. So, superoxide radical, which then produces your hydrogen peroxide. Now, your hydrogen peroxide, we would like to stop. Magpo-produce kasi siya ng hydroxyl free radical. And this hydroxyl free radical will cause oxidative damage to lipids, proteins, and DNA. So we want to stop this process. Now, in order to stop this process, kailangan mag-act upon ang glutathione peroxidase to convert hydrogen peroxide to catalyze or remove hydrogen peroxide, kailangan natin ng glutathione peroxidase. Now, once kasi na ma-oxidize ang glutathione, you can no longer use it. So, pag na-oxidize na natin, ito yung GSSG, oxidized glutathione. Once that is oxidized class, you can no longer use that to convert hydrogen peroxide or catalyze hydrogen peroxide into water. So what we need to do is to recycle it. So i-regenerate siya ng cell. And this can be done by the use of a glutathione reductase. So glutathione reductase, and as you can see class, it needs NADPH. So this process requires NADPH to reproduce glutathione reductase. And if we have glutathione reductase, we can produce reduced glutathione, which can be used by glutathione peroxidase to catalyze hydrogen peroxide. Ang gusto natin itong pathway na to, not this pathway which causes damage. Now here, dito papasok ang glucose 6-phosphate dehydrogenase, yung G6PD. So this G6PD is needed also to produce this NADPH. Okay? Glutathione reductase requires NADPH. So glucose 6-phosphate to 6-phosphogluconolactone. So we have glucose 6-phosphate dehydrogenase, which will produce also your NADPH. So dyan siya papasok class. So if we don't have this reduced glutathione, our hydrogen peroxide will be converted as hydroxyl free radical, which will cause now our damage. Kaya kailangan natin ng reduced glutathione. Now, if we're going to correlate this in a disease that causes this one class, we have what we call your glucose 6-phosphate dehydrogenase deficiency, kaya G6PD deficiency. So the most common disease producing enzyme abnormality in humans is G6PD. In fact, common siya, pero hindi naman siya fatal. We just have to avoid the drugs that can trigger or the instances that can trigger a G6PD deficiency. So it involves decreased NADPH in RBCs and decreased activity of glutathione reductase, causing free radicals and peroxidase to accumulate. So I hope na-remember niyo yung pinakita nating diagram in the previous slide. So what is the presentation? We see hemolytic anemia because these radicals, free radicals, are not being prevented sa pagproduce niya, nasisira. Our red blood cells are lysed. So we have hemolytic anemia. So after oxidative stress, this may happen due to oxidative stress, that is a trigger, due to poor RBC defense against oxidizing agents. And what are our precipitating factors? So these are the things that we need to avoid if your child has a G6PD. So infection, this is the most common precipitating factor. Drugs such as sulfonamides, primaquine, chloramphenicol, and sa diet natin yung faba beans. Okay, so in pathology class, I correlate din natin, we have what we call your Heinz bodies, which are altered hemoglobin that precipitates within the red blood cells. So this is how it will look like under the microscope. We also have what we call bite cells. So bakit bite? Parang nakagatan sila, di ba? So abnormally shaped RBCs due to phagocytic removal of Heinz bodies in the spleen. So we can see here the Heinz bodies are peripherally located. So what happens is there is a phagocytic removal that occurs in the spleen, and in phagocytosis they remove only the Heinz bodies. That's why it appears like bite cells. Okay, now, in the next slides, I'm just gonna give you an idea, class, what are the drugs to avoid in a G6PD deficiency. So, in my experience as a doctor, ito kailangan meron ka sa cellphone mo na copy. Kasi, for example, once na may magpa-check up sa'yo na bata, and the mother tells you that their child is with G6PD deficiency, you have to have a list of this for you to identify what are the drugs that causes hemolysis, and you have to avoid them. You have to find another drug that can treat the symptom of the child without having to use these drugs. Okay, so we have antihelmintics that may cause definite risk of hemolysis, antibiotics, antimalarials, analgesics that are have possible risk of hemolysis, then antibiotics, anticonvulsant, antidiabetic, antidote, antihistamines, and antihypertensives. In short, class, you have to make sure that if you have a child with G6PD deficiency, you have to also do things to avoid infection kasi marami ring gamot na hindi pwede sa kanya. So, yan. Next function is what we call Cytochrome P450 monooxygenase system. So, this class in here, in the Cytochrome P450, alam natin sa liver ito, di ba? So, a supply of NADPH is critical for the liver microsomal cytochrome P450 monooxygenase system. So this is a major pathway, class. This is a pathway for the hydroxylation of aromatic and aliphatic compounds such as steroids, alcohols, and many drugs. So yung metabolism ng mga drugs na to are in this major pathway, and if walang NADPH which is required, it will cause so these oxidations will detoxify the drugs. Therefore, if NADPH is not available, then the detoxification of these drugs, are steroids, alcohols, and many other drugs, is compromised. So it actually converts them into soluble forms that are excretable by the kidney. Meaning, pag walang NADPH, cytochrome P450 monooxygenase system cannot convert these drugs into a more soluble form to be able for the kidney to excrete it. So what will happen? It will damage the kidneys as well. Okay. Next, we have oxygen-dependent bactericidal mechanism of WBCs. Ito maganda itong i-discuss. So we have here, when we talk about this, we talk about phagocytosis of WBC. And yan, oxygen-dependent bactericidal mechanism. So dito papasok yung chronic granulomatous disease na sakit. So explain muna natin. So first, diba, nagkakaroon tayo ng phagocytosis. This is the bacteria. And we are going to form, or nagkakaroon tayo ng phagocytosis by the white blood cells. This is the engulfment of bacteria or any foreign body. So it could be neutrophils and monocytes that causes this. So it uses both oxygen-dependent and oxygen-independent mechanism for killing bacteria. So first, let's discuss oxygen-dependent. So when we talk about oxygen-dependent, it uses myeloperoxidase system and another system that involves generation of oxygen-derived free radicals. Okay? Yun yung oxygen-dependent. When we talk about oxygen-independent class, it utilizes changes in the pH, in the phagolysosome and lysosomal enzymes to destroy the pathogen. So pH naman kapag oxygen-independent. So MPO system is the most potent system. So itong MPO is myeloperoxidase. This is an oxygen-dependent system. So what happens after phagocytosis? So na-engulf na yung ating bacteria. So what happens after that? Nakikita natin there is an oxygen. Sabi kasi natin this is an oxygen-dependent mechanism. So we have oxygen. What's the first needed factor here? We have NADPH. So NADPH oxidase, okay, in leukocyte cells converts molecular oxygen from surrounding tissue to peroxide. O, diba? We need NADPH. Important siya in order for us to kill the bacteria. Ang end goal natin dito is to kill the bacteria. Imagine, class, if we do not have NADPH or sufficient NADPH, it will not move up to this portion. So what happens is, oxygen now is converted into superoxide. Okay, superoxide. Now, superoxide will be converted into hydrogen peroxide by superoxide dismutase. Okay, number 2, sabi niya, di ba? So, we have here in number 2, superoxide dismutase. So, superoxide, which is represented by O2, tapos may minus sign, will be converted to hydrogen peroxide. And remember what we told you about hydrogen peroxide, class? Di ba? It should be converted into water by your glutathione peroxidase. So we have here reduced glutathione. Okay, gagamitin natin yung reduced glutathione. Now this hydrogen peroxide, okay, So, ang next step dito is, in the presence of a myeloperoxidase, number 3, okay, so, in the presence of myeloperoxidase, which is a lysosomal enzyme present within the phagolysosome, peroxide now plus chloride ions are converted into hypochlorous acid. So, peroxide plus chlorine or chloride ions. So, if we have here myeloperoxidase and if it is present, it will convert hydrogen peroxide plus chloride into what we call your hypochlorite. And take note class, our hypochlorite is present in also our bleach solution or is the major component of a bleach solution. Now take note, excess peroxide will then be neutralized by glutathione peroxidase. Now, what happens after this class? Magkakaroon na tayo ngayon ng tinatawag nating respiratory burst. Bakit? Ano ibig sabihin ng respiratory burst? It will now kill the bacteria. Okay? It will kill the bacteria. Now, I want you to imagine, what if in the first process pa lang, after ng engulfment, in-engulf na natin yung bacteria, walang NADPH to convert oxygen to superoxide. So, walang superoxide, walang hydrogen peroxide, which will be, together with the chloride, converted to hypochloric or into hypochlorite, which is a major component of bleach. Will the bacteria be killed? No. What will happen? Andyan lang yung bacteria. Engulf lang ng engulf, pero yung bacteria nandyan lang, hindi namamatay. So, what will be the manifestation? This now will lead into what we call your chronic granulomatous disease, and it will appear this way. The white blood cells continues to engulf the bacteria, but because of the deficiency in NADPH oxidase, it cannot kill the bacteria through the oxygen-dependent pathway. So what happens? It cannot convert molecular oxygen into superoxide in leukocytes and use the respiratory burst to kill the bacteria. So since we cannot kill the bacteria, what happens is we have a severe, persistent, and chronic pyogenic infections caused by catalase-positive bacteria. Hindi yan namamatay. For example, this is the white blood cell. It engulfs a bacteria. So we have now what we call a phagosome. Now, if there is no defect, for example, we have abundant NADPH, we kill the bacteria by the respiratory burst. But if, for example, it is defective because we don't have NADPH, we only form, the microbes will survive. What we form are what we call your granuloma formation. And we do not want that, okay? So, last, another function of NADPH is the synthesis of our nitric oxide. Okay, the synthesis of our nitric oxide. So here, in the last slide, we're going to talk about how is the pentose phosphate pathway being regulated. So how do we know if we need to stop producing NADPH already? So take note class, NADPH, which is generated in the oxidative branch, remember, diba, sa oxidative phase, ito nare-regenerate, of the pathway, can feed back and inhibit the pathway. So glucose 6-phosphate then can be utilized for glycolysis. So for example, in the oxidative phase, our glucose 6-phosphate will be converted into 6-phosphogluconolactone and later on will produce pentose phosphates. Now, once that we produce NADPH, this will automatically send signal in the pentose phosphate pathway that we already have enough NADPH. Therefore, yung glucose 6-phosphate can now be used for other pathways such as that of the glycolysis to produce ATP. Okay? So, in summary class, I hope ma-remember natin kung para saan ang pentose phosphate pathway. So, we have to remember what is it for, where does it occur, and what are its products, and what is the substrate. And that there are two phases, which is the oxidative phase. It is also the irreversible phase. And we have the other one, the non-oxidative phase, which is the reversible phase. So I hope, class, that this simple lecture, we were able to understand ano nga ba yung pentose phosphate pathway and what is its purpose. So having said that, I would like to thank everyone for your attention, and I hope that marami tayong natutunan in this lecture. So thank you and God bless.